Okay. Well, good evening, everybody. My name is Ilya Nemenman. Usually, I'm a professor of physics and biology at Emory University in Atlanta. But today, I'm here on behalf of the Aspen Center for Physics, and I'm here to welcome you to our first virtual Heinz Pagels lectures of 2021, of the 2021 season. Today's speaker is Professor Nic Vega from Emory. However, before I turn the floor to Nic, I will say a few words about the names that I just mentioned, the Aspen Center for Physics, the Pagels lecturers, and Professor Vega themselves. So first, the Aspen Center for Physics. Every summer, some 500 leading scientists, leading physicists from across the globe assemble at the center, each for a few weeks at a time. The center is an iconic institution for us, for physicists, with a scientific impact which is far beyond its small budget. It's an incubator, it's a place where we all brainstorm, collaborate, meet other scientists, some of whom serendipitously become very important in our professional life. As a personal example, I've been coming to Aspen for 16 years now, time flies, and every visit has been magical, resulting in multiple new collaborations, multiple new papers, new understanding of things that I thought I had known before, new ideas, new friendships. Some of my most impactful work would not have existed if not for the center. And it's fair to say, I think, that I'm far from unique. People who visit keep coming back again and again for all the same reasons. Why is ACP so impactful? Well, I don't know. It could be the mountain air, gorgeous Colorado scenery, but most likely it's because the ACP climate, which shelters us from all of the distractions of everyday life, and it's built around opportunities for new interaction, for fresh activities, in-depth discussions, where we can just think and intensely sit and intensely think, focused on important question that we're trying to solve. Let ideas fly around, fertilize engaged minds, and so on. We come here stuck on a scientific problem, often we leave having this problem solved, but with 5 new ones in our minds. And this is probably the most exciting thing in the life of a scientist. So we're very thankful to the National Science Foundation, which partially supports the center as an investment in the nation's scientific base, and we hope that ACP will continue to change physics for years to come. The second thing that I need to explain is the lecture series. The lecture series that you are attending is named after Heinz Pagels, who was a longtime participant at the center. He was a professor at Rockefeller University in New York City, a leading figure in particle physics, a distinguished educator, author of popular science books, president of International League for Human Rights. His work on chaos theory inspired the character of Ian Malcolm in Jurassic Park book and movies. He died tragically in a climbing accident on Pyramid Peak not far from here in 1988, and his friends at ACP have decided that naming the public lecture series for him would be a fitting tribute to his life. So before introducing this evening speaker, let me also mention that the next event in the series is a lecture next week on June 24th, when Ida Al-Khadra from University of Illinois at Urbana-Champaign will talk about the recent physics news that made front pages. It's certain unexpected measurement of magnetic properties of an elementary particle called muon. These things don't come very frequently. And so please join us next week as well. If you want to be on the mailing list for the future events, I will post the mail address in the chat later on, and you can just send an email to Patty Fox at that email, and she'll add you to the mailing list. So finally, tonight's speaker is Nic Vega, assistant professor of biology and physics at Emory University. Nic got her PhD at Boston University, where they studied how bacteria become tolerant to antibiotics. Nic later worked at MIT in the Physics of Living Systems group, studying interactions of microbes in microbial communities. A few years ago, we were lucky to recruit Nic at Emory, where the Vega Lab now studies formation of and function of microbial communities that live inside the gut of a host, in particular a worm called C. elegans. Nic is one of the world-leading researchers in the emerging field of biophysics of host microbiome interactions. And this actually makes me a bit sad, since we asked Nic to talk today not about their exciting work, but about something else, and that something else is COVID-19. It's fair to say that COVID has been the biggest disruptor in our society in recent memory. Clinicians, scientists, including biologists, bioengineers, and physicists have all mobilized to understand it, to solve it, and delivered amazing vaccines in a very short period of time. ACP physicists have also been involved. In fact, the center right now is running a workshop on COVID, which we're attending in person thanks to the vaccines. But as you all know, a lot of people in the country do not understand the disease, they don't understand the numbers associated with it, how scary it is and how not scary it is, and they don't understand how the new vaccines work, and that's why the vaccine adoption has been rather low. And so Nic generously agreed to explain to all of us what scientists have learned about COVID-19 over the past year and how the community has created these vaccines and how they work. So before I give the stage to Nic, let me also point out that there will be plenty of time for questions at the end of the talk. And you will be able to ask these questions by raising your hand, and then I will call on you. But if you have an urgent question that can't wait the end of the talk, then please don't interrupt, but instead type your question in the chat, and I will relay the questions to Nic. So please welcome Nic Vega now.
Great, thank you so much, Ilya. Can you hear me okay? All right, excellent. I'm going to try to share over. And let's make sure. Got this. All right. All right, thank you, Ilya, for the great introduction. As Ilya said, I'm a researcher at Emory. I do not actually work on COVID. I'm here instead wearing my interdisciplinary hat to give you, as an educated, intelligent, primarily non-specialist audience, the tools and the information that you need to have a comprehensive picture of what's going on in the world, what the situation is with SARS-CoV2, our current state of knowledge, and the progress of vaccines and what this all means for the future. So we're going to start at the top. Introduction to the virus of the hour. What is a coronavirus? Zero order, this is a virus. Viruses have been described to me as bad news in a crunchy protein code, and that's essentially accurate. A virus is information. It's information that wants to make more of itself, and it's a parasite. It's going to use you to get there. Specifically, this is a coronavirus. Coronaviruses have a known structure and structured anatomy, and what these viruses are made of is going to tell us a lot about what they do. So this is a single-stranded plus sense RNA virus. It's a single strand of RNA with a poly A tail that looks a lot like a messenger RNA, albeit a very long one. It's encapsulated. This RNA molecule is decorated with a helix of nucleocapsid protein, and that's going to give the genome some shape, and it's going to protect it from the RNase, the RNA degrading enzymes that are super abundant in the world. Around this encapsulated virus, you have a lipid bilayer, which has been stolen from the host and decorated liberally with viral proteins, including the star protein here, spike glycoprotein. This is spike is a category of proteins. These are proteins that are stuck into a membrane, and they present a receptor binding domain, which is going to recognize a receptor and lead to interactions with another cell. This is important for the life cycle of the virus. Basically, the anatomy that it has is going to give us its function. So during entry to the cell, spike proteins, associates with this ACE2 receptor, the surface receptor on the outside of human cells, particularly epithelial cells in the upper respiratory tract. That'll trigger uptake of the virion. When the virion gets into the cell, it's going to merge its membranes, unpack the virus itself, which, as we said, looks like a messenger RNA. It's going to start recruiting ribosomes. We didn't bring any proteins with us. We didn't need to. We're going to make them all in the host cytoplasm. So ribosome proceeds, it's going to make the viral proteins, including the RNA-dependent RNA polymerase. We're an RNA molecule. We need our own polymerase to make copies of ourselves. In addition to mRNAs, we're going to make our way over to the protein manufacturing factories of the cell, over to the endoplasmic reticulum, and we're going to start stealing bits of that. We're going to start stealing it. We're going to decorate it with our own proteins, mature those proteins, use that to encapsulate new copies of the virus, which will then be released into the world. And so all this is going to happen. We have no reason to go into the nucleus at any point. All this is going to happen in the cytoplasm. We're going to use the factories of the host cell and our own machinery to make more of us. And so I've been saying a lot about spike. See, this is a really important protein. This is the target for the mRNA vaccines. This is something that's critical to the function of the coronavirus. So let's talk a little bit about spike and get this. It's going to be very informative in terms of telling us how this protein works, how this virus works. So as we said, the spike glycoprotein and the interaction with the ACE2 receptor is what allows the virus to enter the cell. And this is a specific interaction. It's based on the structure of the spike protein and its capacity to closely match, interact with high affinity with this ACE2 receptor. And it turns out this is really important for determining host range. And this is why we get coronaviruses the way that we do. Most of the coronaviruses that we have that infect humans are zoonotically acquired. They made this transition from things like bats and rats that have an ACE2 receptor structure that's similar to humans, but not quite similar enough. We needed to go through an intermediate host, something that has ACE2 receptors that look a little like bats and rats, look like humans, to allow us to make that next step in evolution. So all of the pandemic coronaviruses from the last 20 years have come across this way. SARS-1 through this fuzzy little guy, palm civet, MERS through camels, now SARS-CoV2 through pangolins. The jury's still a little bit out on that one. And importantly, we know that if we can mount an immune response against the spike protein, if we can make antibodies that bind with high specificity to spike, this is going to have what's called a neutralizing effect. These are going to be neutralizing antibodies that, by preventing spike from binding to the receptor on the cell surface, will prevent infection from happening in the first place. So how do we get from here to a vaccine? We've got the basics. How do we use this to make something useful? And it turns out there's a lot of information that we got from previous coronaviruses that we're going to use, and we're going to learn a few new tricks along the way. So as we said, this is a coronavirus. And coronaviruses have a lot in common. The interaction between the spike protein and the ACE2 receptor is critical for infection by coronavirus. We know that antibodies that we mount against spike are neutralizing. They can prevent infection by preventing this association. And we know from previous pandemics, from the SARS-1 pandemic, that we can mount an anti-spike antibody response. It's a dominant immune response. We can get a lot of it, and that it is protective, and it is neutralizing. These were early days. By the time we get to MERS, we're in a position to do something intelligent about it. We've learned a lot about mRNA over time and how to use mRNA therapeutically. In particular, we know that we have to do some things to an mRNA to turn it into a vaccine candidate. So one of those things is related to the structure of the spike protein itself. This is a membrane embedded protein. We want it to fold correctly. The epitopes that are presented by a spike protein depend on the three-dimensional structure of the spike. So when we present the mRNA to the host cell, we want to make sure that it's going to fold properly, that it's going to give us the right three-dimensional structure so we can mount an antibody response. There's two positions where we can substitute the existing amino acid for a proline. What that's going to do is it's going to make sure that three-dimensional folding happens correctly. So we have the right structure, we can make the right antibodies. Also, anyone who's worked with RNA will tell you it's incredibly fragile. These things are very short-lived, and everything is covered in RNase and RNA degrading enzymes all of the time. We've learned we can do is protect them. By encapsulating them in lipid nanoparticles, we can protect the RNA that we're trying to deliver to the host cell, increase delivery to the host cell, get more mRNA actually into the cells, and this will let us mount a better response. And so we have some data here from proof of concept study using a modified MERS spike protein RNA that we're giving to some mice. And up here, we're looking at neutralizing antibody production. We see that if we inject saline, we don't get anything. As we provide these mice with increasing doses of our 2-proline modified MERS messenger RNA, we get increasing amounts of neutralizing antibodies. That's great. That's what you want to see. That's a dose response. But is it protective? So we can subsequently, after we vaccinate these mice, give them the vaccine candidate, we can expose them to mouse-adapted MERS, to the virus itself. Mice injected with saline are not protected. We have massive viral loads in the lungs. The lowest dose of our mRNA candidate decreases the viral load. It's a little bit protective. And the next two doses were underneath threshold detection. We can't tell these mice have been infected at all. So using the information that we've gotten about how to change an RNA to make a vaccine candidate, we can make something that we can safely deliver into a host cell that'll give us a neutralizing antibody response, which will be protective against infection. And the reason we're able to do this is over this period of time, we learned a great deal about how to use mRNA as a therapeutic. And the basic technology that underlies these therapeutics it dates back to the 60s and 70s. But it wasn't until the 90s that we really started making progress in terms of being able to use it therapeutically. So in the early 90s, we have first demonstrations that we can take RNA, we can inject it into a host, and we can get the protein that was encoded on that RNA. And more that we can get enough of that protein, enough of the correct protein to have an effect. In this case, we're correcting vasopressin deficiency in a mouse. A few years later, we can deliver a protein that is an antigen, in this case, an anti-cancer antigen, mounting an immune response to the cancer in the rodent body. Now it's starting to look viable. A few years later, we have our first clinical trials. And this is for an ex vivo application. We can take dendritic cells out of the body, treat them with RNA, with mRNA, put them back in so they can perform a task. A few more years down the road, and we're in vivo. We have a human applied cancer immunotherapy where we're trying to mount an immune response against a human cancer. This is personalized medicine. Not until a few years later, we're starting to actually seriously put forward mRNA vaccine candidates for an infectious disease. So as of about 2014, there's a reasonable diversity of mRNA therapeutic applications, most of which are ex vivo. We take something out of the host, we take host cells out, we give them mRNA to change what they do, we put them back in. We have this personalized medicine cancer immunotherapy, which is available. It is extant, but it's not something you're going to get at CVS. For that to happen, enter COVID. Now we're going to use mRNA vaccine development technology to take the fastest path from disease to a vaccine in history. And to understand how we got there, we need to talk a little bit about how clinical trials usually work for a vaccine. A lot of preclinical data, we need a decent amount of data to submit for approval to go into a clinical trial. When we get to clinical trial approval, we have this kind of canonical three-phase structure. Phase one is primarily safety. These are very small studies. Once we've cleared safety, and just do the same thing in human as it does in the mouse, we can reapply for permission to go to phase two, which is going to be a larger study, better safety data, more efficacy data. Once we clear phase two, we can submit for phase three, and this is going to be your big randomized study. You have placebo groups, you have treatment groups, and you want here, what you're looking at here is basically the execution of a power analysis. We need enough events in our placebo group and in our treatment group to be able to accurately calculate the efficacy of our treatment with a sufficiently narrow margin of confidence to say that we're above some predetermined thresholds. We also need this to be large enough that we can detect rare adverse effects on the order of 1 in 1,000, and we need enough data per person, 2 months for emergency approval, 6 months for full approval, and we'll talk about why, to confirm that we have assessed safety. Then we can go for full approval. If we get it, we can go to production. This is going to take a while. In the COVID timeline, we're going to be able to cut this down very substantially. Part of this is because mRNA candidates are very, very quick to develop. Famously, the time from the availability of the first SARS-CoV2 genome sequence to the in silico design of the mRNA candidate that became the Moderna vaccine was less than 24 hours. But we still have to test it. We're going to be able to draw on a lot of this pre-existing information. We have a lot of prior development, a lot of prior information from SARS, from MERS that we're going to be able to pull from to shorten this timeline. And when we get into clinical trials, we're going to be able to condense this by substituting personal attention for red tape and by trading money for time. So we'll be able to compress the timeline. A lot of the time we're paying money to do this. We'll be able to shorten things in regulatory sense by getting pre-approval to expand trials. So we have a phase one trial with a pre-approved safety endpoint. If we clear it, we can expand that trial to a combined phase one, phase two, rather than going back in between. In phase three study, the math doesn't change. We still need enough events to give us the power to detect our signal and give us our confidence interval. But we're going to have some advantages in that it's a global pandemic. We have an extremely large susceptible population. That's going to make this easy to enroll. And while we're doing phase three, we're going to do what's called production at risk. We're going to start large scale manufacture. If we don't clear phase three, we have to set all this money on fire, but we're willing to do that. And finally, what may have saved the most time overall is that nothing waited on a desk at any point. Everything was evaluated as highest priority. For perspective, This is a traditional vaccine study. This was a stage 3 clinical for Gardasil, the HPV vaccine. This is, as they are, basically an execution of a power analysis. We have a pre-specified number of endpoints that's going to give us the power to say that we have efficacy against our primary target, which in this case is cervical cancer. Along the way, we're going to collect a lot of secondary data on prevention of infections overall. Now, the problem with HPV is that people get a lot of HPV and mostly they don't know they had it. So we need to target this at a susceptible population that's old enough to be sexually active, but young enough to plausibly not have been exposed yet. So they went 16 to 24. They have to have a cervix so we can screen for cervical cancer. And we have to follow them for a long time because it takes a while to develop precancerous cells. So this is a 4-year study with rolling enrollment over the first year plus. And we ended up with 11,000 people in vaccine and control groups, with a calculated efficacy against infection and a nice narrow confidence interval. By comparison, this was the clinical trial for the Pfizer COVID-19 mRNA vaccine. We're wheels to the ground in July of 2020. We published the first stage of the data in December. Between July 27 and November 14, less than 4 months, we're going to screen, randomize, and treat 43,500 people. It's more than 4 times as large as the Gardasil study. And most of this seems to have happened in the first essentially 6 weeks, because as of October 9th, we have 2 months of safety data for 38,000 of them. This is a phenomenal amount of person hours that had to go into getting these data. And we don't have hours, so we're doing it in people, over 152 sites worldwide. And based on this data set, we're able to get our vaccine efficacy point estimate, our nice narrow credible interval, and say with a high level of confidence that we're above our threshold for efficacy. The math doesn't work differently. We need the same data. We need the same data size, but we can trade money for time, and that's exactly what happened. So this was real fast. Some of the things that were fast about this are going to be fast in the future. We're probably going to see a lot more mRNA vaccines going forward, and mRNA is faster to develop. The other things that were true, especially the dump trucks of money that we're willing to set on fire over the development of this vaccine, may be less true. Probably not going to be quite this fast the next time around. So there's some really interesting myths and misconceptions out there about the mRNA vaccines. One of my favorite of which is, can vaccine mRNA change my DNA? And I really like this one because this is a case where the physics is particularly illuminating. So to start thinking about this, we have to think about what the mRNA vaccine actually is and what it's doing. So as you said, we have a gene. It's the spike protein gene that we've modified to make it usable as a vaccine candidate. Protein substitutions, a few other things. We're going to put this onto a plasmid. We're going to put it in a bioreactor. We're going to do this cell-free in vitro transcription, which is going to give us our product. We're going to clean that up. We're going to package it into our lipid nanoparticles to prevent it from being degraded by RNases and get it into the cell. Once it's in the cell, it'll unpack. The RNA will start recruiting ribosomes, get translated into our modified protein with the right epitopes, which we can then present to trigger the immune response. Now, this all has to happen relatively quickly because, as we said, mRNA is very fragile. It doesn't last very long. The native coronavirus RNA has a half-life about 2 to 3 hours. This one we've modified a little bit, increased the stability, but still within 24 hours, your original bolus of RNA is going to decay into nothing. You don't have very long to do this. The thing about what's going to happen in this, we have to think a little bit also about what mRNA actually does in eukaryotic cell. So in the cells of your body, this is your nucleus. You keep your DNA here. And because your DNA is here, all the machinery for creating and processing your RNA is going to have to be in here. But all of your protein preparation materials out here in the cytoplasm. So the RNA is going to go this way. It's going to get exported to the cytoplasm, the ER, to make use of the protein preparation machinery. Now, a lot of viruses, a lot of RNA viruses, they don't need any of this stuff. They're going to stay out here in the cytoplasm. They're going to use this machinery. Some viruses do want to get in here, DNA viruses, but if you want to get into the nucleus, it turns out you need a key. And that's because your cells are not in the business of just letting anything into the nucleus. You need to be targeted. Proteins that have a nuclear localization motif can get into the nucleus by interacting with importance at the nuclear pores. And this nuclear pore is why this is going to be such an interesting problem. Nuclear pore. This is the guardian of the nucleus. Pore itself is a circular symmetric protein complex, and there is a central channel, 425 angstroms, 42.5 nanometers in diameter, through which we can pass things. But this isn't an open pore. This is a pore that's crowded with filamentous proteins called FG-NUPs. And what these things do is they'll bind to these targeted proteins, these nuclear transport factor proteins, and shepherd them gently through the nuclear pore complex from one side of the nuclear membrane to the other. Anything that is not targeted is going to be excluded. And this gel has a very narrow mesh radius, effective radius about 5.2 nanometers. Anything large, so small stuff can pass through, water, ATP, macromolecules are going to be kept out unless they have the key. And we can actually see this happening if you have an electron microscope. So we're looking here at electron microscope images of the boundary between a nucleus and the cytoplasm. We've got the nuclear membrane here in white, and this dotted line is the exclusion zone formed by the FG-NUPs as they extend into the cytoplasm. And these little black dots are a protein called GFP that we've made this cell produce. Now in A, B, and C, this is just a regular GFP. It's not targeted, it doesn't have the key. It's got no way of actively getting into the nucleus. And we can see there's a lot of GFP kind of around the edges of the FG-NUP barrier. They're being blocked, but they're also inside the nucleus. They're all over the cytoplasm. They're here, they're everywhere. They're diffusing. If we add a tag to these things, if we give them a tag that's the key to the nucleus, if we give them the nuclear code, that's going to dramatically change the picture. All of a sudden, these little black dots or GFPs, we're just going to round them up like ducklings and we're going to usher them right on through. It's remarkably efficient. And the reason we can do this with GFP is because GFP is tiny. It's a very small protein. It's about 3 nanometers this way, 4 nanometers this way. We've got a 5 nanometer pore. It's going to slow down a little bit, but it's going to be able to diffuse through. Our mRNA, by contrast, this is a long polymer. And we can get some insight into why this is going to be a problem by doing some back of the envelope calculations of a similarly sized average mRNA molecule. It's about 300 nanometers long, so it's too wide to fit in this way. If it was linear, we could fit through this way, but mRNAs aren't lines. They're squishy polymers with hydrogen sticking out. They form hairpins, they form secondary structures. And for an mRNA of about this size, we expect that blob of secondary structure to have an effective diameter to be equivalent to a sphere of the radius of about 40 nanometers. That's large enough that it's going to have a hard time diffusing through, even if this was an open channel, which it isn't. We're not actively targeted. It's going to be a hard job getting into the nucleus. We're going to have a really hard time getting through. But let's say by some chance we do. We're still single-stranded RNA. For single-stranded RNA to get integrated into the genome, which is double-stranded DNA, we have to take that single-stranded RNA, turn into a double-stranded DNA product, and integrate it somehow. Now, there are enzymes in the nucleus that can perform each of these tasks. Reverse transcriptases can take single-stranded RNA templates and reverse transcribe it, create a double-stranded DNA product. There are host reverse transcriptases, TERT used for maintenance of telomerases. There are retrotransposons, mobile elements in the genome that might have their own enzymes. If there's a retrovirus, there might be a viral reverse transcriptase. But none of these are coronavirus. None of them. These are enzymes. Enzymes work by recognizing structures and binding specifically to those structures. You have to have the right shape, you have to be the right size, you have to have the right physical properties to be recognized by these enzymes. There's none of these enzymes have any recognition capacity for the coronavirus genome, let alone the spike protein specifically, and none of these have any known activity on mRNAs. But let's say you could. Let's say you somehow managed to get reverse transcribed. Now you have the same problem again. There are enzymes that can patch together 2 pieces of double-stranded DNA, but they're specific. They're structurally specific, and for nucleotide recognition proteins, that means they're sequence specific. So we have enzymes that we could potentially use, but none of them have any specificity. So we have essentially three impossible things in a row that need to happen. And look, I'm a scientist. I have a hard time assigning anything a probability of 0. It's a big world. Each of these things individually is, however, going to be a hilariously low frequency event. For all of them to happen together in the less than 24 hours that you have before your mRNA decays into nothing, I'm not holding my breath. All right, so another misconception about these vaccines is about the future. Are we going to need updates? Are we going to need to get yearly vaccines or boosters like we do for flu? And the answer is a little bit complicated. Coronavirus isn't flu, but to understand why and what that means about what a booster structure might look like in the future, we have to talk about one of my favorite things, which is mutation. Viruses mutate like absolute crazy. Part of the reason for that is the machinery that they use to replicate themselves. Now, you and I, or lower eukaryotes like amoeba, or even bacteria, these double-stranded DNA genome organisms have double-stranded DNA-dependent polymerases, and most of these have proofreading mechanisms that are relatively good. So you have a protein on a squishy polymer trying to incorporate squishy polymers. Mistakes are made. If there's a proofreading mechanism on your polymerase, it can recognize when it makes a mistake, when it puts in the wrong base. You can go back, take it off, try it again, and that's going to decrease your mutation rate. Double-stranded DNA viruses, a lot of these use our double-stranded DNA-dependent polymerases. They'll go into the nucleus, they'll steal the host enzymes. So they have access to this proofreading mechanism, but not to the other proofreading mechanisms that we use on our own genomes. So they have a lower mutation rate, but still relatively high. These guys can't use our polymerases. And viral polymerases, as a general rule, are incredibly error prone. We have a genome size of about 10 to the 4 bases, 10,000 bases in the whole genome. And each copy of an RNA viral genome can have an expected value of 1 to 2 mutations, which is insane. That is a tremendous amount of mutation. However, it's not always true. Nitivirales, which is the order of viruses that includes coronaviruses, has an RNA-dependent RNA polymerase that does have some proofreading capacity. The error rate of this polymerase is about 10 times lower than we see in other RNA viruses. That means the rate of mutations is lower. It's part of why they can have such big genomes. It's also part of why we tend to see smaller levels of antigen variation in coronaviruses as compared especially to other RNA viruses. And that's shown here. Each of these circles is a different virus. And each of these circles has a diameter that's a measure of essentially the surface glycoprotein gene sequence diversity that we see in a virus of this kind. So it's a measure. So spike is a surface glycoprotein. This is antigen variation. The bigger it is, the more variation we see in these surface glycoprotein sequences. And so we've got our coronaviruses down here in red, and we can see a lot of these have pretty small dots. We don't see a lot of variation. These other dots are colored depending on whether or not we have a vaccine for them. So down here, these small dots, these are blue. We have vaccines. We have vaccines that are good, usually better than 90% efficacy. These problem children, HIV, no vaccine. Hep C, no vaccine. Influenza, we have a vaccine, but we have to change it every year to keep up with circulating diversity. And even then, we don't do a very good job. It's one of the few cases where it's easier to hit a smaller target. There's so much variation in these viruses that it's just hard to keep up with the amount of variation that they generate. Now down here, notice SARS-CoV2 is a very, very, very small dot. Part of that is because it hasn't been around very long. It's also true that this dot is bigger now than it was when this diagram was created. We'll talk about that in a minute. So if that's not the problem, what is? Well, it's that immunity to coronaviruses just doesn't last forever. We know from studies in other related coronaviruses, these endemic coronaviruses that cause common cold, you can get reinfected with the same strain 6 to 12 months. Response to SARS and MERS seems to be somewhat longer, 2 years, 3 years. COVID-19 hasn't been around long enough for us to actually know how long immunity lasts. We have some priors from the data we do have. We know we don't see a lot of reinfections in convalescent cases in the first 6 to 8 months. We have some more specific data from some of the big data sets that are starting to come out. There's a big Denmark data set where we see 75 to 85% protection, efficacy within the first 7 months, somewhat lower if you're older. For vaccines, again, they haven't been around that long. We have a couple of data sets that are 6 to 9 months of data, protection lasts at least 6 to 9 months. We'll know more as time goes forward. Literally, we don't have sufficient longevity of data to answer this question yet. time, in fact, will tell. But let's go back for a second to that tiny red dot. Anyone who's been paying any attention knows that there are variants now. There are variants of COVID, and there's a lot of concern about these variants. And there's a lot of questions about what this is going to mean for the future of the pandemic, for the future of vaccine efficacy. And along in those lines, there's good news and there's bad news, but they're both going to take a little bit of explaining. First, just to get a picture of what we're dealing with, it is completely accurate to say at this point, we are dealing with a landscape of variants. Now, we can see even over the last couple of months is the US data, we can see the disappearance of some of the older lineages and the rise of some of these new variants of interest, variants of concern, including this B117, this Brazilian variant, which is known to be very highly transmissible. Variants like P1, which are known to have some immune escape capacity, and increasingly variants with more mutations like Delta. It's gotten a lot of attention in the news. This one's having a field day in India right now. It's showing up in the UK very extensively. As compared to the Wuhan wild type, there's between 13 and 17 total mutations, depending on what sequence you use. In particular, there's these 4 spike mutations that are associated with increases in transmissibility, infectivity, and with immune evasion. So how concerned should we be? Well, good news first. Based on what we know so far, the neutralizing antibodies that we produce after exposure to the wild type spike protein still have protective capacity. They do still have neutralization activity against the variants that we've been able to test so far. We've got some new data set from the UK where Delta has been for longer. So we have some reasonable efficacy data against the Delta variant here. And this is for the Pfizer vaccine. People have received 1 dose. So in the clinical trial, again, this was June last year. This is a different landscape, different variants. Even B117 had barely started to come onto the scene at that point. And after 1 dose, we saw about 82% efficacy against infections as cases. Against B117, we're seeing about 51%, which is kind of marginal. Against Delta, 1 dose, you get about 33%. That's insufficient. 2 doses, things look much better. The clinical trial, we saw this 95% efficacy. B117, we don't see a substantial decrease. It looks essentially the same. If we look at Delta, we do see a decrease, but it's not nearly as substantial. This vaccine is still performing very well in terms of preventing cases, in terms of preventing infections, even against the Delta variant. So what's going on? How do we get data like this? To understand this, we have to remember what we learned about neutralizing antibodies. So a neutralizing antibody, as we said, is an antibody that's going to bind to the spike protein. And this is going to be a topological fit. You have a 3-dimensional structure interacting with a 3-dimensional structure. If this is a tight fit, you have good binding efficiency, you're able to very easily recruit your antibodies to your spike protein. This is going to effectively prevent receptor binding, and thus could prevent infection from happening. If we start to have mismatches, if we change the structure of the spike protein, the way that changes the 3-dimensional structure so that these antibodies no longer bind quite as effectively, that's going to decrease the affinity of the antibodies that we have to the antigen. Now, that's not great, but it's also not necessarily the end of the world, because if we still have affinity, if we still have binding of the antibody to the antigen, we can kind of compensate for this by adding more antibody. So we're going to need more antibody to perform the same amount of neutralization. But if we have enough antibody to do it, we can still neutralize this virus. There's some evidence, at least on a population-wide level, that this is the case, that neutralizing antibody production is going to be a metric that's associated with efficacy. And we can see this if we compare across vaccines, for example. The more antibody you produce, the more neutralizing antibody you produce, the more effective your vaccine is, and the lower risk you are at infection relative to a placebo group. However, what we don't know, this is very important, is how much neutralization activity you need to have to provide protection at an individual level. That is not currently defined. We have some data, but we don't have smoking guns. We do not actually know where this level is. But we do have some information that's going to be relevant once we have a better estimate of this threshold, which is first, we know going in that you have a better antibody response with 2 doses than with 1 dose. And we know that antibody response varies from person to person. As we can see that this happens across studies, is very robust. We can see it very clearly here. So we have here a set of data where here we've got total antibody production, total antibody concentration in the serum of patients. Each of these dots is a person. It's a person, we start at baseline, they're going to have 1 dose and then 2 doses of the Pfizer vaccine. Green dots are people who had no known prior infection. Purple dots are people who had a prior infection, who had previously been infected with COVID. And we can see if we just look at the no prior infection population, again, each of these dots is 1 person. We've got kind of a big bunch of people. We've got maybe some people who are asymptomatic, had COVID, didn't know it. And we've got this big bunch of people who have basically no capacity, basically no neutralization capacity prior to receiving the vaccine. After 1 dose, we have this very dramatic increase in neutralization capacity. Go from basically 1 log to 7 logs of neutralizing antibodies per mil. That's a 10 to the 6-fold, millionfold increase. That's great. But there's also a lot of variation between individuals. Even if we look at the bulk here, we've got about 1,000-fold difference between people at the top of the pack, people at the bottom of the pack, and we've got some people who seem to have mounted no response whatsoever. Going from 1st dose to 2nd dose, we're going to increase that median by about another 1,000-fold. And we're going to tighten the pack up a little bit. Even again, between the top of the pack and the bottom of the pack and the bulk here, that's about 100-fold difference between people who had a very strong antibody response, people who had an okay antibody response, and some people, again, who seem to have no real response whatsoever. So what we have here is a working model where neutralization capacity is protective. The amount of neutralization capacity that you are producing as an individual is going to be what determines whether or not you're able to neutralize a given dose of virus. And that's going to be a function of the antibodies you're producing, the match to the virus you're defending against, and the amount of antibody you're producing in the first place. Again, we're missing a critical piece of information here. We're missing that correlative protection. We do not know how much neutralization capacity you need to be minimally protected. What we do have a pretty good idea on is that this isn't a linear response. If we're comparing against, say, wild type, you may have 100-fold variation in individuals that have very strong response, individuals that have a low to moderate okay response, but if everyone's above that correlative protection threshold, doesn't matter where you are in that pack, you're protected. Got this big plateau. Then we've got some people who mounted a suboptimal response. These are people who are maybe not fully protected against infection, but they are protected against severe disease. They may get sick, but they're not going to hospital, they're not going to die. And a very small number of people who are not going to mount a sufficient immune response. These people are essentially still susceptible. If we have a variant of concern, if we have a variant that's an escape mutant that has a change in its response to these antibodies, you can have the exact same antibody response, but the neutralization capacity that's provided by those antibodies is going to go down because your affinity for the spike protein is decreased. So now we have maybe a smaller percentage of the population that is fully protected, more people that are semi-susceptible, protected against severe disease, but we might get cases, and some people who are potentially not still susceptible. What we don't know is what's going to happen in the future. We don't know what future escape mutants are going to look like. This is very hard to predict. And there's a lot of opinions on what this could potentially look like as to whether we expect to see in the near future escape mutants that are going to essentially evade vaccine immunity or whether we're not. I can't tell you what's going to happen. I don't know what's true. But what is the case is if this is not gotten under control and on a global level, we're going to find out. And that's a situation that I would prefer to avoid. Now, for me, for someone with my training, this is probably the single greatest argument for vaccination on an individual level. Mutations are generated during reproduction and selected for in evolutionary time, selected for in generation time. The fewer infections we have, The fewer generations we feed this thing, the less opportunity you're going to have for one of these things to arise and start to become dominant, and the better chance we're going to have to keep this thing under control. So, summary. The data that we have so far indicate that these vaccines are a tremendous achievement. They're highly effective, they're very safe, and even in the face of variants, they seem to be performing extremely well. The real world efficacy is high. We're seeing consistently that these vaccines are performing probably better than anyone expected them to in real life. There are some things that we were afraid were maybe going to happen that haven't happened. We thought that one option was that we're preventing cases, but we're increasing the pool of asymptomatic infections, which will still be infectious. That doesn't seem to be the case. It seems like vaccination is very good at preventing asymptomatic infections as well. For people who do manage to get infected, these are patients who'd received one dose, they have partial protection. They're still protected against severe disease to a great extent, and these infections have lower viral load, which we know is correlated with transmissibility, so they're likely to be less transmissible as well. And in terms of the side effects of the COVID vaccine, mostly it seems to be a very safe vaccine. These side effects are mostly mild, especially in comparison to, we're not talking about long COVID here, but just symptoms that persist after the acute phase of COVID. Numerical majority of patients have things like clinical fatigue, headache, et cetera, lasting weeks post-infection. By contrast, the side effects for the vaccine, some of them are similar. They tend to be more mild and they resolve much more quickly. So if I'm vaccinated, I got everything in the pack here. It was a rough couple of days, but it didn't last very long. It's okay within a few days. And in terms of longer term efficacy, in terms of delayed side effects, I mentioned middle of this talk that if we're looking for emergency approval, we need 2 months of data. And that's because in all previous cases of vaccination delayed side effects, those appeared within about 8 weeks after vaccination. So one notable case, the influenza vaccine in 1976 had Guillain-Barré syndrome, which is an ascending paralysis that involved the muscles of breathing as a rare side effect. It's not good. This is also a side effect of influenza infection itself. It occurs 17 times more frequently after natural infection. These occurred within 8 weeks after vaccination, and this really only happened with this one flu vaccine. The effects we're seeing now with myocarditis in young men seem to fall into this category. It's a side effect that is very rare. It's rare enough that we could not have seen it in the clinical trials. It is a naturally occurring side effect of COVID infection as well. In convalescent COVID patients in the same patient population, young adult males or late adolescent males, the rate of symptomatic myocarditis is about 0.3%. Post-vaccine, it's about 30 per million. It's about 100-fold lower. It's not always a side effect of the illness. There's a couple of cases where side effect of actually the vaccine, thrombocythemia post-MMR vaccines, another very rare side effect. That is a case where it's not a side effect of the disease, was related to the adjuvant. Again, mostly between 1 and 3 weeks after vaccination, occasionally up to 8 weeks. There are delayed side effects post-vaccination, but these aren't side effects that turn up months or years later. These are side effects that happen very quickly or not at all. So in summary, viruses are fascinating. COVID's a fascinating disease. We've learned a lot from it. I wish we didn't have to. The mRNA vaccines are really extraordinary. And I think people don't understand sufficiently how much information went into this, how much we understand about mRNA, how much information went into this technology in the background, and how promising this technology is for the future. So thank you, everyone. Be safe out there, and I'll take any questions you might have at this time.
Thank you, Nic, very much. And I'm going to clap for everybody, because everybody else is muted, I guess. Now, the way that we're going to do it again is if you want to ask a question, you raise your hand and then I'll call on you, and I'm going to monitor who has their hands raised. Let's see, so far. Okay, Barbara, would you please unmute yourself and just and ask the question?
Hello, this is Barbara and Michael. First of all, thank you for all the energy and thought that went into your presentation. It was fabulous. My wife and I were vaccinated in February, and we're planning. This is a very personal question, by the way. That was our second shot, the Pfizer vaccine.
No, Moderna.
Or Moderna, I'm sorry. Yeah, we're going to get it right. And we're planning a trip to Belize in November, December. Should we be concerned about the expiration of effectiveness of the vaccine? And will our doctor be able to speak knowledgeably about the situation?
I hope that in the next couple of months, your doctor will be able to speak knowledgeably about the situation. I think we're at the point now where we simply don't have longevity of data to really answer that question. We do know that there is a decay in the neutralizing antibody titers over time. There's a half-life of, it seems to be about 60, 65 days. And that seems to be true for vaccines as well as convalescent neutralizing antibodies. But again, we're missing this really critical piece of information, which is how much neutralizing capacity do you need? And until we have that information, until we actually have a better idea of what that is, I don't know that there is actually an answer to that question. Hopefully in the next couple of months, we will have better answers to that. At the moment, unfortunately, I don't know that we do.
Well, thank you for, again, all the energy that you put into this.
Okay. Please, Benedict Callan, if you can please unmute yourself and ask a question.
Thank you for that talk. That was fascinating. I had a question that I had read at some point in the past about the capacity of coronaviruses to actually do some immune evasion by, I think, tamping down on the production of cytokines. And I was wondering if you could talk about that, you know, what is it that the coronavirus actually does in the body that actually helps it evade the immune system? Do we know anything about what's happening between the interaction between the virus and the human immune system?
All right, so I'll preface this by saying I am not a virologist. I'm not an expert on viral biology. I'm not an expert on COVID biology. It's relatively common for viruses to do this. A lot of viruses have accessory proteins that in some way interact with the immune system and interfere with immune responses. This is really common. Some viruses are really great at it, like HIV is amazing at it. Coronavirus is good. Coronavirus has some accessory proteins that do seem to have some interaction with the immune system that do decrease immune responses. And that may be part of why we see actually better antibody production with the vaccines than we do with the actual coronavirus. In terms of the specifics, honestly, you're going to get a way better answer from some of the actual specialists who are at Aspen right now, people who have studied this in more detail, who know more about these proteins, who know more about the specific interaction with the immune system. It's a fascinating topic, but again, I don't have the specialist background to do it the justice that it deserves. These are fascinating organisms. They do fascinating things with these tiny genomes. Talk to an actual specialist about that. They'll be able to give you a way better answer.
Thank you. Okay, do we have any more questions from the audience?
So I think we have Arthur Thompson, his hand up, I can see.
Okay.
Yes, my question is, are there any medical conditions a person might have that would argue against getting either the Moderna or the Pfizer vaccine?
Oh, geez. So I know there are some officially listed counterindications that, again, this is something where I'm going to default to definitely talk to your doctor about this because your doctor is going to know your medical history much better and is going to have a better handle on, like, in your specific case, is this something that is counterindicated. Broadly speaking, it seems to be relatively safe. I know that there's been some questions about whether it's safe for people who have different kinds of immunocompromise to get the vaccines. In most cases, it seems like the answer is yes. But again, like if you have an immunocompromised situation, this is something you want to talk to your doctor about. Say there are people in my family who have things like sarcoidosis, who have MS, who have received the vaccine safely. Their doctors had no issues with it. But the specifics are something that you would want to talk to your physician about. Broadly, they seem very safe, but again, I'm the wrong kind of doctor. I don't want to actually give medical advice. So talk to your physician specifically. But if you don't have something that's listed, if it's not a case where something that is specifically listed as a contraindication, chances are it's probably okay.
Thank you.
Well, let's see if there are any more questions in the room. Okay, there is one more, please. I think it's Barbara again. Barbara.
Yes, this time it is Barbara. Now, I would think, and I've heard, that if you are taking drugs that lower your immune reaction, that could be a really big problem in getting the vaccine to be effective.
Yeah, so this is something that immune systems are complicated. A lot depends on specifically what you are taking, specifically what it does to your immune system, and specifically how that would interact with the immunogenic potential, with your ability to acquire a new epitope presented and actually mature an immune response against that. Again, it's a very complicated topic. It's going to come down, again, specifically, what are you taking? Why are you taking it? And that's something that I want to say talk to a doctor, but I'm not sure. Talk to a specialist on that one, because that is actually going to become a very complicated question. Something there's a lot of active interest in that. There's a lot of active research, and you really want to talk to someone who has the medical knowledge and is up to date on the literature, because things are changing real fast in terms of what we know. So again, I don't think there is a general answer to that one. Find a specialist who really likes reading papers, and that's who you want to talk to about that.
Thank you. The next one, please, Jay Warren. You can unmute yourself and ask a question.
Great presentation. Thank you very much. The question I have is, you described in terms of understanding the durability of the efficacy of the vaccine is more, you understand the half-life of the neutralizing activity, but you're yet to appreciate the threshold that's necessary for providing a durable, immunization from the virus itself. So I'm very familiar with conventional clinical studies like the one you described for safety and efficacy, but I am unable to appreciate what kind of studies would be required to do this kind of durability threshold, and whether you do that in animal models or whether you do that in humans. Seems like human would be necessary to make the final assessment, but I'm trying to kind of guess what those studies might look like, and rather than guess, I thought I'd ask an expert. Yeah,
it's part of the reason we don't have these data is that this is going to be hard to do. So in a mouse model, the smoking gun, what you really would want to do is to have something like a population of mice that you've treated with that you vaccinated, where you can measure the neutralization potential for individual mice, expose them to the same bolus of infectious disease, and correlate, see what is actually the relationship? What is the functional relationship between your neutralizing antibody level on an individual basis and your probability of becoming infected at a given bolus? That's hard to do in people. For human data, it's going to be correlative, mostly because the studies that would be required would be phenomenally unethical and prohibitively expensive, even if they weren't. So for human data, essentially, mostly I think what we're looking at is we're going to get better quality and more data of what happens to the immune response over time, what's happening in populations over time, and then in those populations, what is happening to infection rates as we observe this decay in neutralization capacity. Again, it's very difficult to do this on an individual basis because you have to be tracking individuals over time, and that's very expensive in people. Getting the N required to be really, really hard. But the mouse study, I think, is doable. To the best of my knowledge, it has not actually been done yet.
Well, challenge trials have been discussed. I think, whether those are ethical are really questionable, but they were discussed at the outset of this, if you recall, actually all over the world, less than the United States. I mean, that would actually answer the question, but certainly ethically questionable.
Yeah, I'm not sure what getting approval for that would look like.
Okay, I think we're going to take the last question from Linda Hoffman, and then that will be the last question for today.
Hi, yes, this is Connor. Linda is my mom. So I apologize if I missed this at the beginning, but I've heard you discussing that the mRNA can only last for about 24 hours at maximum, you said. So it seems like you would make the proteins for those 24 hours and then it would be done. Maybe this is just basic biology, but how does it keep making the right proteins to help stop the coronavirus?
It doesn't have to. It only has to make enough protein to mount the immune response. So what you have is about a 24-hour period where the mRNA is still kind of there. The burst of proteins are longer lived than mRNAs. You've got a couple of days, seems like about 3 days of protein levels. And that's sufficient time for those to mount you're going to have that initial inflammatory period. That's going to recruit your antigen presenting cells. They're going to pick up the antigens. They're going to start showing you your adaptive immune system, and your adaptive immune system is going to adapt. You don't need weeks and weeks of protein. What you need is sufficient levels of protein, sufficient bursts of protein and immune response to present that to the adaptive immune system to allow it to start making antibodies. That's all you need.
Okay, so the proteins, go ahead.
Sorry, that's why there was no need to make this. You can make mRNAs that have some proliferative capacity. Apparently, I don't know how you do it, but apparently you can. This didn't need to be one.
Okay, got you. So the proteins kind of prime the immune system in a way.
Yes.
Okay, thank you.
Yeah, they present that epitope, that three-dimensional structure, and that's what you learn against. That's all that needs to be true. Now, it helps to have that second dose. Part of why the second dose helps, it's going to help refine your antibodies, and that's going to help boost your antibody production capacity. That's why we do this 3 weeks apart. So you have the first phase of the adaptive immune response. It's going to give you that first round of immune cells. Then we're going to wake them back up. We're going to say, hey, let's do better. And that's how you get that full immunity.
Would the second dose have the same mRNA, so it would produce the same proteins, or different?
Same one. So currently. Now, there's a lot of discussion about, are we going to have boosters in the future that might have a modified mRNA or modified mixtures of mRNAs to increase capacity to specifically attack some of these escape mutants that we're seeing. The one that we have right now is the same RNA. It's a wild type.
Thank you. That was a fantastic lecture.
Thank you.
Okay, we're going to end at this point. So what I would like to ask everybody to do, please unmute yourself and let's thank Nic. All of us just clap, which is what we would do if we were sitting in the audience. Okay, thank you very much. Thank you for coming. Thank you, Nic, very much.
Thank you so much. Thank you.